Imagine for a moment that your body holds this invisible blueprint, right? A genetic code, so incredibly intricate, it dictates so much about who you are. Everything from, say, the precise color of your eyes to how your immune system fights things off, and maybe even your predisposition for certain health conditions. What happens when there's a tiny typo in that blueprint? Or, a missing page, maybe an entire chapter out of order that just throws everything off balance. Today, we're embarking on a really fascinating deep dive into the world of molecular diagnosis of genetic disease. And this isn't just about finding problems after they've already shown up. It's really about unlocking the power to act proactively. It's about understanding those subtle, sometimes microscopic shifts in our genetic code, which, well, they pave the way for a whole new era of personalized health. Preventative care, even.
That's absolutely right. The field of molecular diagnostics has, well, it's fundamentally reshaped how we approach genetic health. It's been a revolution, really. For many inherited genetic disorders, while a permanent cure might still be down the road, the ability to accurately identify and characterize these conditions using molecular tools, that's immensely powerful. Think about the proactive measures it allows for. Things like carrier detection for couples planning a family, letting them make truly informed choices, or facilitating guided prenatal decisions, supporting things like pre-implantation genetic diagnosis, which can actually prevent some severe conditions from ever being passed on. These sophisticated tools, especially when you pair them with solid genetic counseling, they really represent our best approach right now for preventing recurrence or transmission to future generations. And what's fascinating here is how over the past few decades, we've seen this diverse array of techniques pop up, each one offering different levels of resolution, scope, cost, all things we'll dig into today.
Okay, so our mission for this deep dive is basically to give you a clear, concise shortcut to being well-informed about these really powerful diagnostic tools. We'll navigate their applications, which are surprisingly far-reaching, unravel the methods, but, without getting too bogged down in the jargon, I promise. We'll also talk about some of their current limitations and look ahead to what's on the exciting horizon of this field. And yeah, we'll try to do it with some surprising facts and maybe just enough humor to keep you hooked. Sound good?
Sounds like a plan. It's a fascinating area.
Right. So when most of us think about genetic tests, our minds often jump straight to inherited diseases, don't they? Conditions passed down through families. But I have to ask, how far does the power of these molecular techniques really go beyond that? Is it just for those rare, specific genetic conditions, or is the impact, much broader across healthcare?
That's an excellent question and a really crucial one. The truth is, the utility of molecular diagnostics has just exploded. It's gone way beyond just classic inherited conditions. It now touches on critical aspects of healthcare in pretty much every specialty. We're really seeing a genuine transformation across the whole spectrum of human health, moving us towards much more precise, personalized medicine.
Okay, so give us some examples. Where else are these techniques making a difference?
Right. Well, let's take cancer genetics.
Mm.
Molecular methods are absolutely fundamental there now. It's not just about saying, yes, this person has cancer. We use these tools to diagnose specific cancer types with incredible precision. That often guides the actual treatment choice. For instance, finding an EGFR mutation in someone's lung cancer. That tells us they'll likely respond well to a targeted therapy, one that specifically blocks that mutated protein.
Instead of just general chemo.
Exactly. Which usually means fewer side effects and often better outcomes. These techniques also let us monitor how well treatment is working, look for disease progression, and even detect tiny amounts of leftover disease after treatment, what we call minimal residual disease. That's crucial for preventing relapse. It's a complete shift from one-size-fits-all cancer care.
Wow. Okay, that's huge in cancer. What else?
Then there's pharmacogenetics. This is really at the forefront of personalized medicine. Imagine being able to predict pretty accurately how you specifically will respond to a drug before you even take it, just by looking at your genetic makeup.
That sounds amazing. Less trial and error.
Precisely. It minimizes those nasty adverse reactions, which can be serious, even life-threatening, and it maximizes how well the medication actually works for you. A classic example is the blood thinner warfarin. The right dose varies hugely between people. Get it wrong, and you risk dangerous bleeding or clots. Pharmacogenetics lets doctors tailor the starting dose based on a patient's CYP2C9 and VKORC1 genes. That improves safety and effectiveness right from the get-go.
Okay, I see. So tailoring drugs to our genes.
Exactly. Then there's carrier screening.
Yeah.
Hugely important, especially for family planning. It lets individuals, couples, often before they even conceive, understand if they carry a gene for a recessive condition.
So conditions that might not affect them, but could affect their kids if both parents are carriers?
Precisely. This empowers them to make really informed reproductive decisions. Maybe that's IVF with genetic testing, maybe using donor sperm or eggs, or just being prepared. It shifts the whole conversation from finding out after the fact to proactive planning.
That makes a lot of sense. And related to that.
Related to that is predictive testing. This identifies genetic predispositions to diseases before symptoms even show up. This opens the door to really powerful proactive strategies. Think about families with a strong history of breast or ovarian cancer. Testing for BRCA1 and BRCA2 mutations identifies those at much higher risk. They can then consider things like more frequent screening, preventative surgeries, lifestyle changes.
So getting ahead of the curve years before the disease might even start.
Exactly. It's a chance to fundamentally change their health trajectory.
Okay. Inherited conditions, cancer, drug responses, planning families, predicting future risk. What else is there?
Well, stepping outside human genetics for a moment, these techniques are massively important in infectious disease diagnosis. They allow for super rapid, incredibly accurate identification of pathogens, bacteria, viruses, fungi, parasites.
Like the PCR tests we all became familiar with recently.
Exactly like that. That was molecular diagnostics identifying the SARS-CoV-2 virus with high sensitivity. These methods also detect antimicrobial resistance, a huge global problem helping doctors choose the right antibiotic. And they're essential for epidemiology, letting public health track and contain outbreaks quickly.
Okay, so infections too, that's a really broad scope already. Are there even newer areas?
Oh, absolutely. The horizon just keeps expanding. We're seeing exciting fields like nutrigenomics.
Nutrigenomics, like
Yeah.
nutrition and genes.
Yep. Using your genetic info to get personalized dietary advice, moving beyond generic advice to what actually optimizes your health based on your genes. Or gut microbiomics, analyzing all those trillions of bacteria in your gut to understand their impact on digestion, immunity, even mood.
Wow.
And of course, DNA fingerprinting. That's a cornerstone of forensics. Paternity testing, built entirely on these molecular principles.
Okay, here's where it gets really interesting. It's clear this isn't just for rare diseases anymore. It's touching nearly every part of medicine and even our daily lives.
It really is. And this broad utility highlights a key clinical point. When a genetic disorder is suspected, the right diagnostic approach isn't usually one size fits all. The choice really depends on the suspected level of abnormality,
Yeah.
what kind of change are we looking for? So you might use a broad genetic test if the diagnosis is really unclear, maybe lots of different symptoms. Whereas you'd go for a much more targeted test if you strongly suspect a specific condition. That gives a focused, often faster answer.
Okay, that distinction is important. And it perfectly sets up where we're going next, right? Looking at those different levels of analysis. Exactly. Let's use that library analogy again. Our genetic material is like this vast library. We can look at the whole collection of books. That's like looking at our chromosomes. Or we can zoom in on specific shelves or even individual books. Those are like subchromosomal regions or single genes. And then for the real deep dive, we can get down to individual words or letters within a book. So now we're going to explore the tools that let us read at each of these levels, each designed to solve a different kind of genetic mystery, from the really big stuff down to the tiny details.
Precisely. We can basically group the main diagnostic techniques into three broad categories based on the level of genetic variation they target. First, the chromosomal level, looking for those big, gross abnormalities. Second, the subchromosomal or molecular cytogenetics level, focusing on more targeted or even genome-wide copy number changes. Smaller chunks, basically. And third, the single gene level, getting down to the microscopic details like point mutations or tiny insertions. What's truly fascinating here is how each technique is really cleverly designed to answer a very specific question about that genetic blueprint.
All right, let's start big. The chromosomal level. When people picture genetics, they often think of those X-shaped chromosome diagrams, right? And for looking at that level, we have karyotyping. You called it the classic first look, like getting an aerial view of the whole genetic map.
Karyotyping is absolutely the traditional, foundational method for visualizing and analyzing our whole set of chromosomes. Its main job, its indication, is detecting large chromosomal abnormalities, things you can actually see under a light microscope. We're talking aneuploidies, abnormal numbers of chromosomes. The most famous is trisomy 21, Down syndrome, with that extra copy of chromosome 21. It's also great for spotting large rearrangements, balanced or unbalanced, like translocations, where bits of chromosomes swap places, or big deletions or duplications.
So if a child has, say, multiple birth defects and developmental delays with no clear cause, karyotyping might show a big rearrangement.
Exactly.
It could quickly reveal a large-scale issue explaining that whole picture.
So how does it actually work? How do they get that picture?
Right, well, the technique itself, while established, it's quite meticulous. It involves taking cells, usually lymphocytes from blood, and getting them to grow and divide in the lab. Then, the crucial step is stopping them right in the middle of division in metaphase. That's when the chromosomes are all condensed and visible, like those little X shapes. Then the cells are carefully treated, spread onto glass slides, and stained in a special way. This creates a unique pattern of dark and light bands along each chromosome.
Ah, the G bands, like barcodes for chromosomes.
Precisely, like molecular barcodes. Finally, scientists look under a microscope, identify each chromosome by its size and that unique banding pattern, and meticulously arrange them in order. That order displays the karyotype. It's basically a high-res photo map of your chromosomes.
Okay, but you mentioned limitations. What's the main one?
The crucial limitation is its resolution. It's relatively low resolution. While it's great for seeing the big picture, it just cannot detect small deletions, tiny duplications, or critically single gene mutations. Changes at the letter level are invisible to it.
How small is small?
Well, its resolution is typically around 5 to 10 million base pairs megabases, or engbm. Sometimes down to 3 to 5 millibases if the bands are super clear. But that still means millions of base pairs are below its detection limit. It also needs actively dividing cells, which isn't always practical for every sample type.
And what samples do they usually use?
Peripheral blood is most common, using those lymphocytes. But it can also be done on bone marrow, amniotic fluid, or chorionic villus samples, especially for prenatal diagnosis.
So what does this all mean? It's like seeing the major highways on the map. Are there too many? Are whole sections missing? Great for things like Down syndrome. But it won't show you a small pothole on one specific street, or if a single letter on a street sign is wrong. For that, we need to zoom in.
Exactly. You need different tools for finer details.
Okay, so karyotyping gives us the big picture. But what if the problem is too small for karyotyping, but still involves chunks of DNA, not just single letters? This is where we get more precise, moving to that subchromosomal or molecular cytogenetics level, like going from the satellite view to a detailed street map.
Precisely. This level lets us hunt for smaller, more targeted changes, or even scan the whole genome for these intermediate-sized alterations. Things karyotyping misses, but bigger than single base changes.
So what's the first tool here?
The first one is fluorescence in situ hybridization, or FI. Its main job is detecting specific microdeletions or microduplications. These are small missing or extra DNA bits, maybe involving just a few genes, that are too tiny for karyotyping. Think of DiGeorge syndrome, that's a deletion on chromosome 22q11.2, or Williams syndrome, a deletion on 7q11.23. FI-ish is perfect for those. It's also used for looking at rearrangements near the ends of chromosome subtelomeric regions and for rapidly spotting common aneuploidies even in cells that aren't dividing, which is useful prenatally.
How does FA-ish work? Fluorescence. So it involves glowing.
It does. You design these special DNA probes like tiny searchlights and label them with fluorescent dyes. These probes are designed to bind only to their matching DNA sequence on the chromosome you're interested in. You add these probes to the patient's cells. Where they bind, you see a fluorescent signal under a special microscope. So the presence, absence, or maybe even a different location of those glowing spots tells you if the target region is normal or not.
Okay, but you said targeted. What's the limitation there?
The key limitation is that FISH is highly targeted. You need to have a pretty good idea of what specific region you suspect is involved beforehand. If the genetic issue is somewhere else, your FISH probe won't find it. It's like only shining your flashlight on one specific spot. It also can't detect balanced rearrangements because the amount of the probe DNA doesn't change.
And the resolution. Better than karyotyping.
Oh yes, much better for these specific targets. Typically around 50 to 200,000 base pairs kilobases, or KB, a big jump from karyotyping's millions. Sample types are similar: blood, bone marrow, amniotic fluid, CVS, or tissue.
Okay, so FIH is like a targeted spotlight. What if you suspect a microdeletion or duplication, but you don't know where it might be?
Ah, yes, that leads us perfectly to chromosomal microarray, or CMA. CMA is often a first-line test now, especially for kids with unexplained developmental delay, intellectual disability, autism spectrum disorders, or multiple congenital anomalies.
Why first line?
Because it offers a comprehensive genome-wide scan for those submicroscopic copy number variants, the microdeletions and microduplications that FEH would only catch if you pointed it right at them. CMA can also detect aneuploidies. It's become really indispensable in pediatric genetics. It provides answers for a significant chunk of previously undiagnosed cases.
So how does CMA scan the whole genome like that?
It uses a chip, Basically a small glass slide spotted with millions of tiny DNA probes. Each probe represents a specific region across the entire genome. You take the patient's DNA, label it fluorescently, say with green dye, and wash it over this chip. Sometimes reference DNA, maybe labeled red, is added too. The patient's DNA binds to its matching probes on the chip. Then a scanner measures the fluorescence intensity at each spot. Computer software compares these signals. If there's less patient DNA binding in a region, left green signal, maybe more red if using a reference, it indicates a deletion. More signals suggest a duplication.
So it's comparing the patient's DNA amount across the whole genome to a reference standard.
Essentially, yes. It allows for simultaneous detection of copy number changes genome-wide.
Okay, this sounds like a truly powerful wide net approach, able to scan the entire genetic landscape for these smaller chunks of missing or extra DNA. What can it do?
The main limitation is that CMA cannot detect balanced chromosomal rearrangements, like inversions or balanced translocations.
Why?
Because in those cases, there's no net gain or loss of genetic material, just a rearrangement. The copy number stays normal, so the signal intensity doesn't change. It also can't detect single nucleotide variants, those single-letter changes, or things like methylation abnormalities.
And its resolution compared to karyotyping, or FIS?
Significantly higher than karyotyping for detecting these CNVs genome-wide. It can typically detect changes down to maybe 5 to 50 kilobases in targeted areas, and maybe a few hundred kilobases across the genome backbone, much smaller than karyotyping can see.
Samples. Still blood.
Yep, whole blood is common. Amniotic fluid, or CVS, can also be used, especially prenatally if there are anomalies suggesting a CNV.
You mentioned earlier that interpreting these wide net results can sometimes be tricky.
That's a very perceptive point, yes.
Right.
Because CMA looks everywhere, it can sometimes find variants of uncertain significance, or VUS. These are copy number changes that haven't been definitively linked to disease before. Their clinical impact isn't immediately clear.
So what do you do then?
It becomes a complex puzzle. Clinicians and genetic counselors have to investigate further. Often involves testing the parents to see if it's inherited, checking large databases. It really highlights the need for expert interpretation alongside the tech.
Right. Okay, that makes sense. Now, speaking of screening, especially in pregnancy, there's one we hear a lot about, NIPT, non-invasive prenatal testing.
Yes, NIPT is primarily a prenatal screening test,
Yeah.
mostly for common fetal aneuploidies, trisomy 21, 18, 13, and sex chromosome issues like Turner or Klinefelter syndrome.
Yeah.
Increasingly, it's also screening for some specific microdeletions, and it can be done quite early, usually from 10 weeks gestation.
Non-invasive means just a blood test from the mother.
Exactly, a simple maternal blood draw. The amazing part is that tiny fragments of cell-free fetal DNA, which actually come from the placenta, circulate in the mother's bloodstream. NIPT isolates this cfDNA and sequences it using next-generation sequencing, NGS. By comparing the relative amounts of DNA fragments from different chromosomes, it can infer the fetal chromosome dosage. More chromosome 21 fragments than expected suggests a higher likelihood of trisomy 21.
But you emphasized screening test. Why is that distinction so important?
Critically important. NIPT tells you the likelihood or risk. It doesn't give a definitive diagnosis. A positive NIPT result always needs confirmation with an invasive diagnostic test, like amniocentesis or CVS, which directly samples fetal cells.
So there can be false positives or negatives.
Yes. There can be false positives or false negatives, especially for rarer conditions, microdeletions, or sometimes due to issues like confined placental mosaicism, where the placenta's genetics don't perfectly match the fetus's. Its resolution is basically chromosome level for common aneuploidies. For microdeletions, it's more limited, usually catching ones bigger than 5-7 megabases, and sensitivity varies.
Sample is just maternal blood, often in special tubes.
Correct. Special tubes to preserve that fragile cfDNA. And understanding that screening versus diagnostic difference is absolutely key for anyone considering NIPT. It manages expectations.
Okay, clear. What other techniques are used in that prenatal space, maybe for faster results?
For rapid diagnosis of those same common aneuploidies 13, 18, 21, and sex chromosomes, we have quantitative fluorescent polymerase chain reaction, or QF-PCR. Its speed is its big advantage, especially if an ultrasound flags a potential issue and you need answers quickly.
How does QF-PCR work?
It uses specific markers on the chromosomes called short tandem repeats, or STRs. These are short repeating DNA sequences. You use fluorescently labeled primers to amplify these STRs. Then you measure the amount of the amplified fragments, the alleles. The ratio of the allele peaks tells you the chromosome dosage. Normally, you'd see 2 peaks for a marker on chromosome 21. With trisomy 21, you might see 3 peaks, or 2 peaks where one is twice as high, a 2.1 ratio.
So it's counting chromosome dosage using these markers. Limitations.
The main limitation is that it only detects that limited set of common aneuploidies. It won't find balanced rearrangements, small CNVs, or single gene mutations. Its resolution is chromosome level, based on those specific markers. Samples are typically amniotic fluid or CVS prenatally, though blood can be used postnatally.
Okay, moving on from aneuploidy detection, what about looking for smaller deletions or duplications within specific genes?
Right, for that, a very useful technique is multiplex ligation-dependent probe amplification, or MLPA. MLPA is great for detecting copy number variations, specifically exon-level deletions or duplications in known genes.
Exon level, so parts of a gene.
Exactly. Think of Duchenne muscular dystrophy, where the DMD gene often has deletions or duplications of one or more exons, or spinal muscular atrophy, linked to SMN1 gene deletions. MLPA is excellent for those. It's also good for known microdeletion duplication syndromes, where you know the exact region to target. It gives a more granular look at copy number within a gene than CMA.
How does it achieve that? Ligation dependent. Does that mean joining things?
It does. You design multiple probe sets. Each set has 2 halves that are designed to bind right next to each other on the target DNA sequence within an exon, for example. Only when both halves bind correctly can they be joined together ligated. Then, these ligated probes are amplified by PCR. You measure the amount of each amplified product. More product than normal means a duplication of that target sequence. Less product means a deletion.
Clever, like needing 2 keys to turn simultaneously. Limitations.
It's limited to the known target sequences covered by the probes you design. It can't find single nucleotide variants or balanced rearrangements. Its resolution is excellent for its purpose, exon level or even sub-kilobase resolution within those targeted regions. Sample is typically whole blood.
Is there a variation of MLPA for other types of genetic changes?
Yes, there's methylation-specific MLPA, or MS-MLPA. This is specifically for diagnosing imprinting disorders or conditions caused by abnormal DNA methylation patterns.
Methylation. That's not a change in the DNA sequence, right? It's something on it.
Exactly. It's an epigenetic change. Think of it like a chemical tag, usually on cytosine bases at CpG sites, that doesn't change the underlying A's, T's, C's, and G's, but acts like a switch, turning genes on or off. Disorders like Prader-Willi, Angelman, and Beckwith-Wiedemann syndrome are caused by problems with these methylation patterns, these imprints.
So how does MS-MLPA detect these tags?
It's similar to standard MLPA, but adds a crucial step: digestion with methylation-sensitive restriction enzymes. These enzymes are special. They only cut DNA if a specific CpG site is unmethylated. By comparing results from samples treated with the enzyme versus untreated samples, you can figure out the methylation status of those specific CpG sites.
So it tells you if the switch is on or off at key locations. Same limitations otherwise.
Pretty much. Shares limitations with standard MLPA regarding target specificity and variant types. Its resolution is focused on the methylation status at specific CpG sites. Sample is whole blood again.
Okay, one more in the subchromosomal category. You mentioned something newer, optical genome mapping, OGM.
Ah, yes, OGM. This is a really exciting, more cutting-edge technique. It's indicated for detecting and characterizing large structural variants, the complex stuff like big deletions, duplications, inversions, translocations, insertion, even repeat expansions that are often really difficult to sort out with WGS, microarray, or karyotyping.
So it catches things the others might miss.
Potentially, yes. Especially complex rearrangements. It's proving really useful for undiagnosed rare disease cases where you suspect something complex is going on structurally. And it's becoming important for FFHD, facioscapulohumeral muscular dystrophy, which involves contraction of a big repeat array on chromosome 4. OGM can map that structure well.
How does it work? Optical mapping. Sounds like it visualizes DNA differently.
It does. It uses ultra-long DNA molecules, hundreds of thousands, even millions of base pairs long, much longer than typical sequencing methods handle. These long molecules are fluorescently labeled at specific sequence patterns. Then they're linearized, maybe by flowing through tiny nanochannels, and imaged. Software analyzes the pattern of fluorescent labels along these super long molecules and compares it to a reference map to spot structural variations. It directly visualizes large-scale genomic architecture.
Fascinating. Limitations.
It can't detect single nucleotide variants or small indels. And it needs really high-quality, ultra-long DNA, which can be technically challenging to extract reliably.
Yeah.
It bridges a gap. Its resolution for detecting variations spans from around 500 base pairs up to the megabase scale. It's particularly sensitive for variations spanning tens to hundreds of kilobases.
Sample type needs that long DNA.
Yeah, whole blood is preferred for getting that high molecular weight DNA. Fresh tissue can sometimes work too.
Wow, this sounds like a fascinating new frontier, catching those really complex rearrangements that might hide from other methods, like analyzing the entire page layout and binding of the book, not just the words.
That's a good analogy. It provides a different, complementary view of genome structure, especially for large, complex alterations.
Okay, let's zoom in even further now. We've done chromosomes, subchromosomal chunks. What about the individual words or even letters in our genetic books? This is the single gene level, where we find those really subtle yet often profound changes, where one misplaced letter can mess up the whole instruction.
Indeed. These techniques let us pinpoint incredibly precise changes, often down to a single base pair. And the first one, still considered the gold standard for many things, especially when you know exactly what you're looking for, is Sanger sequencing.
Sanger. That sounds familiar, the original method.
Pretty much the classic method, yes.
Yeah.
Its indication is detecting single nucleotide variants, SNVs. That's a change in just one DNA letter, and small insertions or deletions in bindles, usually less than 50 base pairs or so. Crucially, it's used when you're looking in a known gene or a specific region. It's also very often used to confirm variants found by other, maybe faster but slightly less precise, methods like NGS.
So if you need absolute certainty about one specific spot,
Sanger is your go-to for that. It provides single base pair resolution.
how does it work? How does it read the letters?
It uses special modified DNA building blocks, dideoxynucleotides. When one of these gets incorporated during DNA synthesis in the lab, it stops the process. You run 4 separate reactions, each with a different dideoxynucleotide, A, T, C, G, or nowadays often using fluorescent tags for each base in one reaction. This creates a whole set of DNA fragments, each ending at a specific base. These fragments are then separated by size, usually using capillary electrophoresis, like sorting them through a very fine gel. As they pass a detector, it reads the fluorescent tag on the end of each fragment, allowing you to reconstruct the sequence, letter by letter.
So it literally spells out the DNA sequence. What kinds of disorders is it used for?
Various monogenic disorders where specific mutations are known. For example, confirming known point mutations in the CFTR gene for cystic fibrosis.
And samples.
Whole blood is common. Buccal swabs, cheek swabs, or tissue can also work.
So, if you know exactly where to look, Sanger is your reliable detective for those tiny, crucial changes, like a high-powered magnifying glass for reading every single letter on one specific page. Why still the gold standard with newer tech around?
That's a great point. Even with the power of next-gen sequencing, Sanger remains the gold standard because of its extremely high accuracy for confirming specific known variants. Its low error rate and clear resolution of individual bases make it perfect for validation, or for targeted testing when only one or a few specific changes are suspected. It's precise, robust, and relatively simple for analyzing just one gene or region.
Okay. But what if you don't know exactly where to look? Or you suspect the problem could be in one of many genes, or maybe anywhere?
Ah, right. That's where the power of next-generation sequencing, NGS, really comes into play with whole exome sequencing, WES, and whole genome sequencing, WGS.
Yeah. What's the difference again?
Okay, so the exome, WES, is just the protein-coding parts of our genes, the exons. It's only about 1–2% of the entire genome. But crucially, about 85% of known disease-causing mutations are found within these exons because they directly dictate the protein structure.
So WES focuses on the most likely culprits.
Exactly. It's a broad diagnostic test, great for suspected Mendelian disorders, especially if the symptoms are complex or don't fit a clear pattern, and maybe single gene tests or panels were negative. The technique involves capturing these exonic regions, often using tiny probes like molecular lassos, pulling them out, and then sequencing just those parts using NGS. It's an efficient way to survey the most functionally important regions.
And whole genome sequencing, WGS, that's everything.
That's everything. WGS sequences the entire genome coding regions, non-coding regions, all the DNA between genes, the lot. It's the most comprehensive test, indicated for undiagnosed rare diseases, especially if WES was negative. Maybe the mutation is in a non-coding region that regulates a gene, or it's a complex structural variant, or a repeat expansion that WES might miss.
How does WGS work? Just sequence it all.
Pretty much. The entire genome is fragmented into smaller pieces, adapters are added, and then NGS platforms generate millions, even billions, of short sequence reads. These short reads are then computationally aligned back to a reference human genome, like putting together a massive jigsaw puzzle to identify where the patient's sequence differs.
And resolution for both WES and WGS.
Single base pair resolution within the regions they cover.
Hmm.
So WES reads every letter of the exome, WGS reads every letter of the entire genome.
What kind of disorders are these used for?
Undiagnosed rare Mendelian disorders, complex neurological conditions, really any heterogeneous genetic disease where the answer isn't obvious. They often provide diagnoses for patients who've been on long diagnostic journeys.
Sample is blood again.
Yep, whole blood is standard. Fresh or frozen tissue can also be used. So if we go back to the library analogy, WGS is like reading the entire library, every single book, cover to cover, footnotes, everything. The most complete picture.
And WES?
WES is like focusing intensely on reading just the main chapters of every book of the exons, where most of the critical story and most known errors tend to be. It's more focused, often quicker and cheaper, if you suspect the issue is in a protein coding region.
Got it. Okay, what about those weird stuttering bits of DNA you mentioned, the repeat expansions? Is there a special tool for that?
Yes, there is. For diagnosing triplet repeat expansion disorders, the gold standard is TPPCR, which stands for triplet repeat prime PCR.
Triplet repeat, like Huntington's disease.
Exactly. Huntington's involves a CAG repeat expansion, Fragile X syndrome is a CGG repeat, myotonic dystrophy, a CTG repeat. There are various spinocerebellar ataxias, C9orf72-related ALS, all caused by these abnormal expansions of short DNA sequences.
So how does TPPCR measure these, especially if they get really long?
It uses a really clever primer strategy. One primer binds outside the repeat region. Then there are two priming primers that bind within the repeat sequence itself. This design allows the PCR amplification to kind of walk through or read across the entire repeat region, even when it's massively expanded, which standard PCR sequencing often struggles with. It generates a characteristic ladder-like pattern of PCR products on a gel or capillary trace. The pattern of that ladder tells you if there's an expansion and how big it is.
Like a ruler specifically designed for measuring stutters.
That's a great way to put it. Its resolution is single triplet repeat resolution, so it's very precise for sizing.
The mutations.
It's specific to known repeat sequences. It won't find other mutation types. And for extremely large or complex expansions, sometimes you still need confirmation with an older technique called Southern blot.
Sample.
Whole blood is typical.
This sounds incredibly clever, catching those tricky stuttering sections. It's amazing there's such a specific tool for that kind of genetic glitch.
It is very ingenious. Now, another PCR-based technique, but for a different purpose, is quantitative PCR, or qPCR. qPCR is used for detecting gene dosage or copy number for specific gene regions. It quantifies how much of a particular DNA sequence is present.
So like confirming a deletion or duplication found by CMA, but focusing on one gene?
Precisely. It's often used to confirm or exclude CNVs, or to accurately count the copies of a specific gene. It can also be used to quantify gene expression levels by measuring RNA converted to cDNA, which is useful in some contexts, like detecting gene fusions in cancer, where expression might be altered.
Resolution.
Its resolution isn't base pair level, it's quantitative detection of gene copy number. So it tells you how many copies, not the exact sequence. A key example is quantifying SMN1 gene copies for spinal muscular atrophy diagnosis and prognosis, as copy number correlates with severity. Sample is usually whole blood.
Okay, one last one in this category, something called inversion PCR.
Right, inversion PCR is highly specialized. It's specifically for detecting inversions.
Where a piece of DNA gets flipped around.
Exactly. A segment gets cut out, flipped 180 degrees, and stuck back in. If this happens within a critical gene, it can disrupt it and cause disease. A really important clinical example is in the F8 gene, which codes for factor VIII, essential for blood clotting. Large inversions involving intron 22, or sometimes intron 1, are a common cause of severe hemophilia A.
So how does PCR detect something that's just flipped?
It uses primers designed to bind to sequences that are normally oriented away from each other. But if the inversion has occurred, these primer binding sites are brought close together and oriented correctly towards each other, allowing PCR amplification to happen. So getting a PCR product of a specific size tells you the inversion is present. It's very targeted for specific known inversions.
Primarily for hemophilia A, then.
That's its main clinical use, yes. For those common F8 inversions, sample is whole blood.
Right. We have just navigated through this incredible landscape of current diagnostic tools. From the really broad strokes of chromosomes down to single letters of DNA. It's amazing. But science never stops, does it? What's happening beyond the clinic in research labs? What tools are they using to understand life's deeper mysteries and, shape the future of medicine? This is where it gets really interesting, looking at the tools pushing the boundaries.
You're absolutely right. The research frontier is incredibly dynamic. And these next techniques, while mostly used in research now, are vital. They do two things. First, they help us understand the why behind the diagnosis, the mechanisms of gene regulation, cell processes, how genetic changes actually cause disease. It adds depth to the clinical findings. Second, these research tools are essentially the breeding ground for the next generation of diagnostic and therapeutic tools. This is where future clinical applications are born.
Okay, so what are some of these research powerhouses?
Well, first there's transcriptomics, usually done via RNA sequencing, RNA-seq. Remember, genomics looks at the DNA blueprint. Transcriptomics looks at RNA, the dynamic messages being sent from the DNA to make proteins. It's about gene activity. RNA-seq gives us a global picture of gene expression. Which genes are switched on? How strongly? Are there novel types of RNA messages? Are genes being spliced differently to make different protein versions? Are genes fused together abnormally, like in some cancers? It tells us what's actually happening functionally in a cell or tissue at a specific time.
Are there variations of RNA-seq?
Oh, yes. Two really exciting ones are single-cell RNA sequencing, scRNA-seq, and spatial transcriptomics. scRNA-seq lets us look at gene expression in individual cells. This is huge because tissues are complex mixtures of cell types, and diseases often affect them differently. sRNA-seq reveals that cellular detail. Spatial transcriptomics goes even further. It maps gene expression while keeping the tissue structure intact. You can see where in the tissue certain genes are active. Imagine seeing gene activity patterns within a tumor biopsy. Incredibly powerful for research.
Wow, okay. From the whole tissue down to single cells and even where they are. What else?
Then there's epigenomics analysis.
Yeah.
We touched on methylation with MSMLPA, but epigenomics is broader. It studies those heritable changes in gene expression that don't involve altering the DNA sequence itself. It's about the modifications to DNA or the proteins, histones it's wrapped around.
The software controlling the DNA hardware.
That's a great analogy. It includes things like whole genome bisulfite sequencing, WGBS, to map DNA methylation across the entire genome, identifying all those off switches. ChIP-seq tells us where proteins, like transcription factors that regulate genes, are binding to the DNA. And ATAC-seq maps regions of open chromatin areas of the genome that are physically accessible and ready for genes to be turned on. These are crucial for understanding complex diseases where gene regulation goes awry, cancer, autoimmune diseases, even neurodevelopmental conditions.
Okay, so we've got DNA, RNA, epigenetics. What about the end products, proteins and metabolites?
Exactly. That brings us to proteomics and metabolomics. Proteomics is the large-scale study of proteins. What proteins are present, how many are they modified, who are they interacting with, what are they doing? Proteins are the cell's workhorses, so studying them shows cellular activity directly. Metabolomics studies the small molecules metabolites that are the inputs, intermediates, and outputs of cellular metabolism. Things like sugars, fats, amino acids. They give a real-time snapshot of the cell's physiological state, like checking the chemical readouts of the cellular engine.
So these show what's actually happening functionally downstream from the genes.
Precisely. They provide a more immediate picture of health or disease state.
Is there a way to combine all these different omics?
Yes, and that's a major goal now, multi-omics. This involves integrating data from multiple layers, genomics, transcriptomics, proteomics, metabolomics, epigenomics from the same sample. The aim is to get a holistic, systems-level understanding. How do all these layers interact? How does a change in DNA ripple through RNA, protein, and metabolic networks to cause disease? It's challenging, dealing with all that diverse data, but it holds immense promise for understanding complex biology and disease.
Okay, these omics give us huge amounts of data, but how do researchers confirm if a specific genetic variant they find actually causes a problem?
That's where functional assays come in. These are lab experiments designed to directly test the impact of a variant. Does this specific change actually harm protein function? Does it mess up gene expression? Does it alter how cells behave?
How do they test that?
Several ways. They might use cell line engineering, often with CRISPR gene editing, to introduce the specific variant into cells in a dish and see what happens. Or use induced pluripotent stem cells, iPSCs. These are made from a patient's own cells, like skin cells, and reprogrammed back to an embryonic-like state. Then they can be turned into specific cell types, like neurons, if it's a brain disorder, allowing researchers to study the disease in patient-specific cells. Another tool is using minigene constructs to study how a variant affects RNA splicing in isolation. These assays provide the crucial link between finding a variant and proving it's pathogenic.
Right, the functional proof. Are there other advanced techniques changing the game?
Definitely. One major area is long-read sequencing. Technologies like PacBio, especially HiFi, and Nanopore. Unlike standard NGS, which generates short reads, maybe 150, 300 bases, these sequence very long DNA or RNA molecules, thousands, even hundreds of thousands of bases long.
What's the advantage of reading long stretches?
It's revolutionary for several things. Detecting large structural variants that short reads struggle to span. Resolving complex, repetitive regions of the genome, phasing alleles accurately, figuring out which variants are on which chromosome copy from which parent, and characterizing full-length RNA molecules without assembly. It's opening up parts of the genome that were previously hard to analyze.
And anything else really pushing the boundaries?
Well, there are chromatin conformation capture techniques, like Hi-C. These investigate the 3D structure of the genome inside the cell nucleus. DNA isn't just a line, it folds up in complex ways. These techniques map which distant parts of the DNA physically interact. This is important because DNA folding can bring regulatory elements close to genes they control, even if they're far apart linearly. It adds another layer to understanding gene regulation. So, connecting this back, these research tools, they're not just academic exercises. They're constantly deepening our fundamental understanding of disease. And that knowledge directly fuels the development of the next generation of diagnostics and treatments we'll see in the clinic. They are the engine driving future medical breakthroughs.
It really paints a picture of relentless progress. #tag outro.
So what does this all mean for us, the listeners? We've taken quite a journey today, haven't we? From seeing those big chromosomal outlines with karyotyping, zooming into smaller chunks with CMA and FISH, all the way down to the single-letter precision of Sanger and WGS. And then we even peeked into the future with omics technologies revealing entirely new layers of our biology. It's just a field that's constantly pushing the boundaries of what we can know about ourselves, moving us ever closer to really understanding that blueprint of life.
That's absolutely right. The landscape of molecular diagnostics for genetic disorders just keeps expanding. And we're clearly seeing that shift towards higher resolution, more comprehensive approaches, WGS, maybe OGM starting to enter routine clinical use as the tech gets better and, importantly, costs come down. Ultimately, that means more answers, hopefully faster for more patients, leading to earlier interventions and much more targeted, personalized care. Yeah, imagine a future, maybe not even that far off, where a simple routine test could flag potential genetic predispositions, but also precisely guide health decisions. Diet, medications, everything offering truly personalized wellness tailored right down to your unique genetic code. Of course, that raises some big questions too, doesn't it? What other, maybe profound ethical considerations or societal shifts might come with having such detailed, pervasive genetic knowledge about ourselves and each other? The questions this science raises are honestly as fascinating as the science itself. So we definitely encourage you to continue your own deep dive, maybe explore those ethical implications, or look more into one of the specific technologies that caught your ear today. Thanks so much for joining us for this deep dive.
Thank you. It was a pleasure.